Excellent. Well, thank you very much for the introduction there, Brian. And it's also a very nice complementary set of talks that are being put here together. And certainly the previous talk from Aldebaran removes the need for me to explain the intricacies of how to actually make viral vectors. So thank you very much on that side of things. So as I think has been stated several times in the course of this morning, it's of course a very exciting time for cell and gene therapies. This is a slide I've been sort of doing this talk, or this particular slide I've been showing for about the last year or so. And it seems like every time I have to put it up, I get to update some of the numbers that are on the particular slide. So the most recent number that I had to update was the 4 approved products in the US since 2017. And that was, of course, the approval of Zolgensma from Novartis and Avexis on Friday last week. If you include other approvals, this number around the world, this number, of course, increases as you look at European approvals and things like that. But certainly behind those approved products, we also see a substantial pipeline. So 93 products in phase 3, more than 1,000 products in development around the world, and a phenomenal amount of money being spent in this particular area as well. And indeed, if you there was an MIT study that came out last year that was cited by the FDA to suggest that there would be on the order of 40 gene therapy products on the market in the US by 2022. So very large development efforts going on in this area, and very exciting times in terms of patients and the ability to treat some previously very devastating diseases. But of course, one of the key aspects to this is how do we actually manufacture these particular therapies? And one of the key things that needs to be considered when you think about how to develop these therapies is what is the scale that you really need to manufacture these at? And that is going to determine the technologies that you need in order to invest in order to put these together. So what I'm showing up on the screen here is a relatively simple equation which really determines what the amount of viral vectors that you need to produce per year is. Now, while it's a relatively simple equation, the thinking that has to go into each of the coefficients within this particular equation are actually quite important and very extensive, and really quite fundamental business decisions that need to be made as you're developing your product and your business. So first of all, there's the number of patients per year, and that's of course going to be dictated by the number of patients that suffer from that particular disease. It's also dependent on therapy access and reimbursement, a very hot topic within the cell and gene therapy space, and also your assumed market share within this space as well. And all those are going to determine the number of patients that you have to treat. Obviously, the patient dose is also pretty fundamental to it all. So, and that's going to really depend on what particular therapy you're actually treating. So, a small eye-based disease such as Lux-Turner is going to be dosed in the case of with about 1.5 times 10 to the 11 viral genomes per eye. But then if you're going up to Duchenne muscular dystrophy, you could be talking about something like 10 to the 15 viral genomes. So a log-4 scale of different dosing that's going to be taken into account there. And then there's, of course, also the number of manufacturing runs per year that you're going to want to be able to do as well. So if you're going to be doing very few manufacturing runs per year, that may at first seem quite an attractive prospect. But of course, if you're only doing very few manufacturing runs, then you're probably not going to be very good at making your product. And if you've spent all the money on a large-scale manufacturing facility, then it's also going to be a very inefficient use of your capital investment in that case. On the other hand, if you are doing too many runs per year, then there's going to be a very high-risk supply chain associated with your particular drug. And all of that, these different factors, is going to determine what scale of manufacturing you really want to be getting into. Now, I think many people have put up variations on this particular slide, and certainly viral vector manufacturing has largely been done in an academic setting where each of the different unit operations may not be particularly scalable up to the size that you necessarily want to have done in a GMP setting. So for example, vector production these days is largely done in transient transfection type systems. Purification can a lot of times be done with using ultracentrifugation. And then, of course, there's the analytics associated with the entire manufacturing train as well. And the analytics associated with viral vectors is currently quite challenging, not particularly accurate, and really does, in many cases, limit the ability to manufacture these viruses in a consistent fashion. So now, while there are many people working on more advanced technologies in order to make this more scalable, there are particular things that can be done with existing technologies and connecting existing technologies together to make viral vector manufacturing something that can be taken to GMP in today's settings. So a lot of the challenges associated with current viral vector manufacturing are yield and titer, and also consistency and reproducibility of these particular things. So certainly moving to novel expression systems and novel manufacturing technologies is something that should be done, and something that I'll show you in this particular slide, but in this particular presentation. But also what I'm going to be talking about today is the role of integrated solutions to bring more consistency and more reproducibility to manufacturing of these particular drugs. So something that we're very involved with in Paul Biotech is taking existing conventional processes, so you can see things like flatware type processes, and we can work with you to move those to a bioreactor type of system. Also looking at, rather than ultracentrifugation, looking at implementing things like chromatography and things like that. So there are a variety of unit operations that are available now that can help to industrialize the manufacturing of viral vector technologies. And here I'm just showing you the various technologies that we have at Paul Biotech that can support that, whether you're working with an adherent type of a manufacturing system or more of a suspension type manufacturing system. We can help with those. And there's also a variety of downstream processing technologies as well. And here are a variety of consumables that can help, and also, depending on which particular scale, we usually have the appropriate type of technologies that are involved with all of that too. I'm not going to spend a lot of time in today's talk talking about each of these individual technologies, but rather than that, I'd like to talk about how all of these technologies can be brought together in an overall integrated solution that can be implemented at a GMP scale and really bring about more consistent and reproducible manufacturing. So at Paul Biotech, we've been involved with putting together integrated solutions for both monoclonal antibodies, recombinant proteins, as well as viral vectors for a number of years now. And in fact have done on the order of 25 different projects in this particular space at this point. And this is the overall process that we generally use in order to implement these integrated solutions. The key thing is to understand your particular process and what it looks like now, and then to ensure that we design a solution around that that is able to fit together and is able to be implemented in a consistent fashion. So we generally start over on the left-hand side by really working with you to characterize your particular process and go through a mass balance type of a process to really make sure that we understand, both you and we understand all the particular elements associated with that. Once we figure out we have a common understanding of the process itself, we then go into an overall integrated solutions design. So that can go through overall plant and equipment layout, then looking at process flow, then making sure that we can physically connect the different unit operations associated with that. And so that involves a lot of single-use manifold design. We then discuss with you a variety of automation concepts that we have and how you would like to automate these particular things. Once we've all gone through the overall integrated solution design, we then move to an execution phase where we actually will build equipment and then go through a factory acceptance test. And then we actually go through delivery and implementation of that, so a site acceptance test, and then an overall training and making sure that you're able to use the equipment in the facility on an ongoing basis. So really connecting all of these elements together. So just to step through what each of those particular steps looks like. So this is usually the first stage of a mass balance type of a setup. So this is where we map all of the inputs and outputs from each particular unit operation. And here I show two particular examples, one from an upstream cell culture perspective, where we're looking at a facility to generate on the order of 10 to the 16 viral genomes. And we're using an iCELLis 500 in this particular process. And you can see here where we've mapped in the inoculation of the bioreactor, and then the overall recirculation, down to the lysis particular process, and then that goes through into all the other unit operations after that. We also have mapped here a particular anion exchange membrane chromatography step, where again you can see that we've really looked at all of the inputs and outputs from each particular unit operation. What this allows us to do is it allows us to do a lot of equipment sizing and specification, generate an equipment list, and then also really understand the interconnectivity between those different unit operations as well. And also, probably most importantly, ensures that both you and us have a very complete understanding of how the overall process fits together. Once everybody's agreed and signed off on the mass balance, we then start to lay out the particular plant and equipment. And so there we use a variety of tools. So in this particular case, we have a standard tool, three-dimensional tool, where we can actually map out our particular unit operations. So again, this is the 10 to the 16 viral genome AAV type process, where we've mapped everything out, including from seed train all the way down to sterile filtration at the end, including the upstream processes. You can see we've mapped out the process flow over on that diagram on the right, and really made sure that all of this equipment will fit into the particular footprint that you have available in order to implement this. We also use three-dimensional viewing of this particular technology to make sure that all the equipment helps. This may initially seem like a bit of a gimmick, but actually it really helps significantly to make sure that there is adequate space to move around these particular pieces of equipment. And again, to make sure that if you have particular operators in there, they're able to move around from different pieces of equipment in a seamless fashion. So that we find is a very helpful process in order to help the overall process design. Once we've done that, it also becomes very important to look at a process flow view of this. Obviously, one of the very key and most important aspects is to make sure that we can excuse me, physically connect these different unit operations together. So these process flow diagrams that I show here are very important for doing that. And what going through this particular process helps with is really designing the single-use manifolds that allow you to physically connect these different things. Now, the design of these single-use manifolds is very, very important. We know that a lot of times there is significant product loss that takes place in the downstream processing of viral vectors. A lot of the time the loss of that is due to hold-up volumes and things like that. So by really optimizing the single-use manifold design, we can do things like reduce hold-up volume associated with particular unit operations and drive higher yields through these particular processes as well. We have our Allegro Central Management System, or ACMS system, that allows us to design these single-use manifolds. What we have in there is a lot of pre-validated connections between different operating mechanisms. So for example, looking at different single-use connectors, or different barb type connections. We've pre-validated a whole bunch of tubing types and connectors, which allows us to very quickly and efficiently design these systems together. And it also allows us with a very, very good communication system with you to make sure that we have taken everything into account. These are all, of course, customized to you as well, and allows you to have sign-off on those particular things too. And this is just an example of some of the drawings that come out of that type of system there as well. We've also recently signed an agreement with GCON, so as you know, a lot of times speed to market is very, very important when it comes to viral vector manufacturing. And we've worked with GCON to come up with some pre-existing designs around what viral vector manufacturing may look like. Again, there is no expectation that this will be the exact facility that you will end up going with, but it allows us to be about 70 to 80% there, and should allow for much faster design of your overall process and then much quicker implementation of your manufacturing process after that as well, so getting you to market faster. And of course, what's very, very important in all of this process is ongoing communication between both Paul and yourselves as well. So we make sure that we have a very high level and key project management system that is involved to ensure seamless communication between us all and the overall flow of the project in as seamless fashion as we can get. So overall, hopefully I've given you an idea of the overall Paul Biotech integrated solutions piece in here as well. Something that I haven't mentioned in here as well is also our process development services. So our process development services will also allow you to design the process if you would like to do that as well. And we've actually done that process development services, as I said, for more than 25 clients at this point in the viral vector space as well. So a lot of experience around designing and getting these processes up to speed in this particular context. So again, hopefully that's given you an overview of what we're able to do within Paul Biotech. We've got a very wide range of technologies that's available to be implemented around here. And probably what's more important here is helping you to get your manufacturing facilities up and running as quickly as possible when we know that time to market is very, very important within the gene therapy context. And so that, I'd just like to thank you for your time. I will be available to discuss any and all of these options available, and I look forward to speaking with you, and I wish you a good bio conference after this. Thank you very much.